infrastructures, TreatmentBank, and the Biodiversity Literature Repository. With Plazi, he leads the development of novel means of liberating taxonomic data from the primary literature, including the TaxPub and JATS XML schemas for taxonomic treatments, and more recently, the material citation class in the Darwin Core Standard, which allows for specimens cited in the literature to be linked directly to their occurrence records provided by museums or other sources. He works closely with Pensoft publishers to implement these tools to automate data extraction from the literature, with over 30 journals now making use of them, including phytokeys, zookeys, and African invertebrates. Plazi also publishes taxonomic treatment datasets directly to GBIF, which provide over 80,000 taxon names, which do not exist in any other GBIF datasets. And just to put that in context, GBIF gets taxonomic data from approximately 2 billion published specimen records. So that is quite a notable contribution from Plazi. He currently lives in Switzerland and has lived and worked in the UK, Australia, the USA, Egypt, Iran, and France, due to his better diplomatic half, who has also been one of the reasons that Plazi and many of the advances in access to biodiversity literature have been possible. He is father to 2 sons, neither of whom want to become biologists. He also enjoys nature and is an avid iNaturalist contributor. So thank you very much, Donat, for joining us this afternoon, and over to you.
Thank you very much for these kind words, and well, I'm thrilled to be back in South Africa, even though it's only virtual. I started my scientific life essentially in East Africa, so Africa is like the first love. That's the most important one. So still, it's one of my best times in my life, walking there in the Miombo forest and looking at plants and plant termite interactions. So today I will talk about the rediscovering known biodiversity. So when I was a young guy, a young boy, I liked to go into the forest and just discover things, because we were living next to a big forest. And I also like to go to libraries, because it was an interesting thing to start with one book, and then the book talks about something, and then you go find the next book which talks more about this. So you end up in one corner, literally, and from where he started. When I was at my high school, I had a little project to recreate a double helix, because I was really like to read science books, and I was really interested and intrigued by Watson's double helix. So I had to go, and had the privilege that I could go to a library and had some pocket money, go to bookshops and buy books, and essentially then assembled a model of the DNA. A little bit later, now making a jump, I ended up in New York, and we got some money to do a follow-up on the conventional diversity monitoring. So we thought it should not be just the elephants and the baobab which need to be protected, but there are tons of ants and other insects which should also have its word. So we looked into assembling the world's experts on ants. We developed standard methods for measuring and monitoring biodiversity. And that, in a way, in the mid-90s was the beginning of all what I do today. And still, I would say the few questions you see here are the ones which are really driving us. So you go out, you want to know what taxon is it. Today, you can use Google Lens or iNaturalist, and often it is something, you get a name. Often you don't know, so you have to go still back to keys and to books. But once you have a name, you want to know anything about this taxon. You want to know its distribution. And those are still the questions that drive us in biodiversity, I think. At the same time, we started to digitize. So when I was living in Cairo, I couldn't work in the lab anymore, but I could hire some students to help to digitize in the first round all the reprints I had. And you can see here clearly the impact that had. And I think it's probably one of my biggest impacts in my life. So when I started my PhD, everything was concentrated at Harvard. So everybody working on ants had at some point had to go to Harvard, misuse their zero-copy machines, fill up their suitcases, and travel back home. But not long after we digitized all the 4,500 ant publications, we've seen a phenomenal use of this of these PDFs all around the world. And it was really, I think, the beginning of a world where people realized that how important it is to have PDFs and make them accessible. So when we talk today about open access, it's mainly about getting free access to PDFs. But we essentially are much more interested in what's in publications. We are interested in the data of publications. Like here you see one thing which is in all these publications and nobody knows about is there are millions, hundreds of millions of illustrations. But these illustrations are not just interesting per se, but these illustrations, they are linked. So each of the illustration, you can, from here, you can go click on it, you go to its deposit in our repository, the Biodiversity Literature Repository. From there, you can of course go back to the publication, and often the publication is closed access, so you cannot get the publication, but you have a lot of links inside. So we extracted taxonomic treatments, we extracted the figures you have already seen, and from that, other people can use this data. You can go to GBIF and look at the article. That's the GBIF view of an article, or you can look at specific data on the specific species. So essentially, what this allows is not only to go to an occurrence in GBIF, but also the vice versa. So the interesting thing about this is if you have a point, like an occurrence record in GBIF, you can now actually go back and say, okay, this is part of this species, which is part of this publication, of this treatment, of this publication. So now you have a link, which is given here in the metadata, which is, okay, this occurrence belongs to that. And it's not just the link, which is important, citing like a PDF somewhere you can't access, but it's all the data which is in this publication. I added the PDF to the link to the PDF in the chat, so you can actually go into this in the PDF, and look up all these links, so you can actually follow up this yourself. But there's something more to it. It's not just that we have now a specimen in PDF in GBIF, but it can be reused. So GBIF is reusing the data we liberate, but there are already like 500 publications that actually reuse this data. So essentially, this is what you want. You publish a scientific work, you want to make it to be used by others. And this way now, this is happening in a increasingly big scale. So if you want, what we do is becoming part of the research data lifecycle. That means you collect your data, you do all the analysis, here you publish it, and then we come in, we extract the data because you publish it normally in PDF, we put it into the entire workflow, it goes back to GBIF, and it can reuse that. So this is really a nice example of a working research data life cycle. Of course, I tell you at the end how we could do this much better and how you can do it without engaging us. There's something else which is also interesting. So when you publish a PDF, the PDF can be cited, you have a DOI and can be cited. But if you start to take it apart, you add more links to the PDF. So your data has more exposure. So if you put it to TreatmentBank, you create a second publication, which is essentially metadata and links to data within the publication. You expose the treatment. If you put it on Biodiversity Literature Repository, you have the publication again, as I showed before. You have the figures, you have treatments. If it goes to GBIF, you have even more. So you have a view of the publication you have already seen. You have the figures, so you see the figures, you can click and go back. You have the treatments, you have material citations. And so like there are 4 ways, 4 exposures if you have just a publication which has 1 treatment and 1 figure inside. So if you have, like, say, you have a publication which has 10 figures, 10 treatments, and 30 material citations, it has like 84 entry points now into these publications. So that's another way which is really interesting for publishers. It's interesting for you as an author. So people have find your data much easier. But you can also do more to that. So we kind of talk in our green world, the biodiversity world. So we take a treatment out, and as I showed you already, it goes into GBIF, and then somebody makes distribution models, or we have other tools that are using our data. But we can expose it to the life science community by making it part of PubMed Central. So we can actually bridge the gap between the silo of life sciences and the biodiversity community by adding all our treatments there, so they can use their very extended and very sophisticated text and data mining tools we are still missing in our community. We can also add data to Wikidata, so it becomes more a citizen science exposure. And of course, the important thing now is that Google crawls what we do. So if you go on Google and you look for not so common species, you often end up in our place or in data we liberated. But it's not just the data in itself. A publication has a wealth of citations inside. So if you look like on e-Flora, you see all the implicit or explicit links in publications. So you have a nomenclature section of biology ecology, but then you have references. And references are like this. And if you make them accessible, you can click on the PI on the precision identifier, and you actually can go, in this case, to a treatment, or you can go to a journal, or you can go to a treatment. But if you have immediate access. Similarly, if you look at material citations, they are very rich in data itself. So their material citation includes a link to an institution, there's a specimen code, or it includes collectors. And collectors we could identify and actually then start to make statistics, or actually just make a list and say, who has this guy what has this guy collected? If you go a bit further, and we try to do that, essentially is to look into ways to link all this data in and references in publications to the cited resources. For example, you have a specimen code here. You want to link it to the specimen code and to the actual physical specimen at the end, or at least a digital copy of it. But we also want to do something different. We want to actually go to the one we cite and say, hey, listen, we actually cite your specimen. Here is the source. Here, these are us. So they could embed a link and say, okay, this specimen has been cited in this material citation, in this publication. Another interesting thing is that, like, if you're interested in what people do, there are all different ways where people are exposing publications. So a very direct one is a collector. So a collector collected this specimen, or a collector described a publication. So you have the author, or the person is an authority of a taxonomic name. So you have a person already in 3 different functions. And again, if you actually soon, hopefully, can start linking all these things, or put all these things together through bidirectional links, then we have something really cool in our hands. So this is part of a EU-funded project, Bicycle, which started in May and lasts for 3 years, and essentially brings together the main infrastructures in life sciences, in DISCO, in the specimens, GBIF, then of name, and literature. That means we are involved, and also Biodiversity Heritage Library. But now that you have all this data, you can think of many interesting things. So one I like is, for example, they expose all new species. So you can go on our Plazi website and every morning go and look at new species. You can do that on your mobile phone. Just go on plazi.org and type in and look on the map and click on the green dots, and you actually get back to new species. And I think this is really exciting. But of course, I'm very limited in my ideas, so I'm sure you have many better ideas how to make use of all this liberated data. So how is Plazi liberating data from publications? So first, we make use of something which is really at the base of taxonomy. So when Linneo started in 1758, he is hailed for creating the Latin binomen, like in this case, the honeybee Apis mellifera. So here it's clear, it's the shortage, the L for Linnaeus, and we know if L point is, it's 1758. So we know because we experience, we know. But Linnaeus did something much more clever. He essentially created in his book, the Systema Naturae, the treatments. And these are blocks of text which are dedicated to one particular text and one particular species. Oops. In this case, it's honeybee, so there's a taxonomic name. Then he already has treatment citations. That's he cites previous works. So he says, okay, there's been cited with Swammerdam in this work before. Then he has a statement about distribution. He has a description which uses already kind of a standard vocabulary. And then in this case, that even a section of biology, ecology, where he defines what the bee does, what flower exists. Implicit, this taxonomic treatment is based on the honeybee, which is in the Linnaeus collection. And it is, of course, clear it's part of this book. So we have essentially everything we need to start to extract all this information. So because, and I tell that because we are in a privileged position that our data is very, very nicely structured, whereas like in humanities, it's much more difficult to accomplish something similar. And you've already seen that there's a lot of data in publications. So when you actually look at a publication, it's already a big citation network, even within publications. And if you make this data citable, somebody like GBIF can come in and start to build access to it. And so you can actually go look not just at the PDF as you do as a human, but you can say, okay, I'm interested in a specimen. What do I know about a specimen? So you can start from here and find out, okay, there's a figure published about this. Oh, here's a treatment. Here's a physical specimen. So it opens up very different ways to look at this data. And we do that by collaborating with CERN, the nuclear facility in Switzerland, in the European nuclear facility in Switzerland, who has a huge storage area, and at some point decided we should actually give something back to the small sciences like biodiversity by making them use of by extended knowledge of how to manage big amounts of data. So today, this is called fair data. So it's findable, accessible, interoperable, reusable data. That means you take a figure out, you don't just expose the figure, but you add a lot of metadata to it. So you know from where it is, you know what it is, so you can find it. You add a license, like in this case, it's open. You make it interoperable by adding enough metadata again and make it reusable. So other machines can read that. Essentially, that means you use like JSON or other formats which are widely used. So again, here I have a publication which is closed access. Here is the open form, so the PDF. If you go to us, so in this case, it's a closed access, so you don't see it, it's closed, but you have here the bibliographic references, and there's a lot of metadata. So essentially it says, okay, this is the publication, so you have the DOI, there are some tags, it's published here, and it cites these references, but it also cites materials, figures which have been included, or it cites treatments which have been extracted. And again, for computer people, you can download this entire information in JSON format, so you can actually reuse it very easily. So we talk about digital accessible knowledge. So this is kind of primary data that are both digital and accessible in standard formats and need to be built. They needed to build a digital copy of known biodiversity knowledge accessible for human consumption only. So I refer to what the context of all is, what we do. So we assume, Biodiversity Heritage Library assumes that there are something like 500 million pages of printed records on biodiversity. Maybe they scan 10%, maybe there are 10 to 30% accessible through scientists who make their PDFs, but a huge chunk is not accessible. Furthermore, every day, hundreds of new publications are published. We don't know anything about unless we are like specialists or stumble upon that, but we don't know what's inside, and often we find out, but then end up on a paywall and are not sure whether we should spend like $30 to pay for it or go to sources where we could at some point get them for free. So the target functionality of the digital accessible knowledge is that we can essentially build a digital copy of our known knowledge of biodiversity. It doesn't mean we have to make everything exactly digital accessible knowledge, but it's important everything is in a form that computers can come in and produce that. For example, a treatment has traits inside. You don't have the capacity to work on traits, but the life science community has tools and others have tools to be efficient in mining traits. So if you make them the text accessible, then they have tools and can do something really interesting to that. There are some side effects. So if you look at the gains, so think about digital accessible knowledge. Think about or compare in your mind, if you have a plant name which has sort of an authority and you want to find the publication, you want to find the treatment, how long it takes you to be, especially if you're not domain specialist, to figure out which is the publication and treatment referred to. If you have only mouse click, of course, it's very clear. It's a no-brainer. It's really fast. Digital knowledge avoids errors in translation. So if you publish digital knowledge, it's here. So nobody has to go and make an interpretation. Your data is immediately relevant. So imagine you publish a revision in the morning and like a couple of hours later, you find everything in GBIF and accessible. This is really what you want. What at least I think I want. And I mentioned already, so digital accessible knowledge can be much more widely distributed. And it makes essentially our knowledge first-class digital citizens. This is very important, again, in the context of biodiversity conservation, because you want to have data accessible for a lot of ingenious ideas, how to conserve that. And especially we are interested in to help to support those species for which there is no advocate out there right now. So a lot of species, they're only known by a publication. So these are the long tail of species which probably are going lost first. So we better make sure we can account for it. So to do it, we built the Biodiversity Literature Repository. I mentioned already, that's a collaboration between CERN. Right now, it has 400,000 images, 65 out of 65,000 articles. It has something like 184,000 treatments. We run our own, that's TreatmentBank, that's a processing facility where we have about the same number of articles, but we have 620,000 taxonomic treatments and we have like 1 million material citations. And the difference is because there is a quality control step in between. So we don't just upload everything which came out automatically, but we have like rigorous quality control, a step in between. So we only let things up which are compliant with our standards. And if you compare what we have in TreatmentBank with GBIF, then you see that there are 34,000 treatment article data sets in GBIF. It's about half of what we have. We have they have about half the taxonomic treatments. and they have about half the material citations. So there's a big backlog on our side waiting to get into GBIF. So that's, now you see later why this is happening. Now, you probably asked the question copyright. So saying this is our interpretation and how we follow copyright. So it's not our interpretation, it's the interpretation or the advice from one of the top Swiss copyright lawyers with whom we looked into this. We run workshops and wrote papers about that. And we did that over many years now, and nobody so far challenged any of these things. So we seem pretty confident that this is one way forward. And essentially what it says is, in articles, everything is closed access, remains closed access after 1999. But anything before 2000, we treat as open access. So we don't care, if you want, about whether something is open or not. And if somebody comes, we will deal with that. But so far only, and nobody came and complained. All the articles are obtained by a legal access. So legal means in a Swiss context, which I will not explain completely all of what it means, but essentially we go to a library and get the source, or we go to publishers and get the articles from them. And articles are processed irrespective of access and restrictions. And that's a speciality in Swiss copyright law, that there's an exception in the copyright law which says you can data mine anything if it's for sciences. The figures we extract made available, and we have an agreement with the Swiss Collecting Society who takes care of copyright issues. So if somebody comes and says, no, it's my image, they go to them, and then this collecting society would pay something. It's like if a musician plays, or a radio station plays some music, the radio station will play some money to the artist. And of course, from a scientific point of view, we make all this data citable. So taxonomic treatments, it's the same. So we consider it's data because it's just the language everybody's using. And again, we use, from a scientific conduct, of course, we are interested to cite everything, similar to material citations. So our workflow we installed over the years now essentially goes along the line that we get publications, we decode them, we enhance them, so that's a semantic part. We run data quality. If the data quality is good enough, we create open fair data. That means we upload treatments, figures to Zenodo, to the Biodiversity Literature Repository, and return get the DOIs. Then we provide access. We disseminate data. That means we send a note to GBIF and say, look, here is something new, and GBIF comes and picks it up. Then GBIF installs the articles as treatment article data set, makes a view of the treatment, the occurrences, and then the scientist comes in and starts doing analysis, adds more new data, and writes research publications. And we then pick it up and goes in. So that means increasingly there are occurrences here which have multiple material citations, but this is what you want because each one is a bit different. Each one adds something new to this specimen. So just an overview of the degree of automation. So we need, the collecting is something we need human input. Preparation is to make templates so we can automatically process. Decoding runs completely automated. The enhancement, the semantics need some human input. Quality control is really something where we need human input. And dissemination, again, is completely automatic. That's essentially what I show here. So here is essentially what people like you are important because they can go in and they know whether material citation is properly interpreted or not. But all these parts here can be run by IT specialists. And how digital data is today, you make it once, you make it right, you do it only once. So the data you can access from our interfaces. So we have all open data. you can go in and explore that yourself. So our really goal is to build a big initiative now to liberate and make all scholarly published data about biodiversity discoverable and reusable. So we hope you join us in this endeavor. So we want to build a global community involved in data liberation. I hope some of you will follow that and help us to make it big. But we also work here in Switzerland to build a sustainable infrastructure. So we already have sort of unlimited access for storage and long-term storage from CERN, and we're close to having an agreement to build essentially the entire extraction workflow on open government services. That means we have a long-term production service, and the idea there is that essentially it becomes attached to the normal library, so a journal comes in and automatically will be picked up and processed. You can go and see whether something needs to be curated by a person, and you can do it. And of course, and I'll come back in a minute on that, we need to change our way we publish so we don't have to do all this very complicated stuff. So this is essentially in a visual way. So we want to build this global biodiversity data liberation service. We have to build it essentially once, then we can of course have parallel ones, but that means just we don't have to duplicate anything. One publication processed need not to be processed anymore because it's digital accessible knowledge. But we need input, like from the publisher side, we need input from the Biodiversity Heritage Library, for example, to scan more publications. We need input, domain input in the enhancement, like in the tagging. We also need in the quality control input. So essentially what you're going to see tomorrow is this step of this workflow. So we have programs which help you to go. They show you how to decode, to enhance, and do some data quality of publications. There is a little bit of steep learning curve because we are not used to look at publications this way. And it's some technology behind, but I'm sure a lot of you find this interesting, not least because at the end, you see your product here. And that means also everybody in South Africa has really fantastic access to the data. So tomorrow workshop will be run by Karolina Sokolovic and Felipe Simoes. I'll be in the background, and if you have any technical questions, our IT guy, Guido Sauter, will be there. But he is hiding, and we hope we don't need, because we hope everything is so well done and prepared that we don't really need a technical backup. To end essentially is that what we really need to do is you have to switch from the legacy publication workflow, which is really cumbersome, expensive, to prospective publishing workflow. That means we publish in a way that it goes straight into the Biodiversity Literature Repository. And I put Pensive there because they are, together with us, spearheading this effort. We kind of approach them and talk together how we could do it, but they are the publishers, they're important, they invest money to actually make this happen. And I think really, if you're serious about making data and biodiversity accessible, think about, make sure that you publish somewhere where your data shows up immediately in GBIF, because that's in a way the proof of that it works. So I will not go on in this, so you can download the PDF, and you have all these links inside. Here is an example for completely automated processing. That's one of the really cool things if it happens. So you can follow all the link and see what happens, what can be done. So at the end, I would say thank you for everybody in my team. So I'm only sitting here now and having the pleasure and the honor to talk to you. But of course, I can't do anything without the very big, very skilled people and dedicated people. And we can only advance as well through very generous support through foundations and EU grants and national different like Swiss universities and the National Institutes of Health grants and support for NGOs by Microsoft, Google, DataCamp, and so on. Well, thank you.
Great. Thank you very much, Donat. Wow, I think that was quite a whirlwind tour of everything that Plazi has to offer. That was really good. There was some great insights into it there. All right, so we're going to go to questions now for this session. So can I ask anybody who might have a question, if you can just put your hand up in Zoom here, or you can drop your questions into the chat and we'll pick them up there. So anyone with questions would like to fire them off and we can get started. Okay, Nicola.
Hi, yeah, thanks. Thanks very much, Donat. That's really interesting. I wanted to ask, so, I mean, obviously, probably like most people here, I'm more familiar with the traditional way of publishing in a journal and comes out in a PDF. I was just curious as to what this new way of publishing might actually look like, that you were describing, the one, you know, the one you're working on with PenSoft.
Oh, well, for you as an author, it not necessarily need to change a lot. So you submit a Word document and it goes into the processing. What's changing is that you get a PDF, you get an HTML, but you also get an XML copy. So if you go to PenSoft like VitalKey or Sukeys and look at an article, you can download it as XML. So that's in a way the big change. And the more drastic change is the Biodiversity Data Journal, where you don't publish manuscripts as Word, but you publish into forms. So, for example, you have a description, you copy paste it in a form that is description. But you can also drop a Excel spreadsheet, and you will convert it into in a format that can be submitted automatically to GBIF.
Oh, I like that. I like the idea of forms. Anyone who's reformatted their taxonomic data for different journal requirements, I think will be quite keen on that. It actually sounds like quite an improvement. Yeah, thanks.
All right, have we got any other questions about Plazi? Yeah, I think my question was also just kind of revolving around what the implications would be of shifting to that PenSoft model for authors, but I think that's been answered.
Well, I can only say there are two efforts now on the way. So there are two funded projects. One is the bicycle, which goes along the ARPA writing tool, which makes it more open for everybody. And there is one from CTAF, the European Consortium of Taxonomic Facilities, publishing working group, which got money from the French government to create an XML workflow. So you can see there's some progress on the XML side in the near future. And the other point is that what we do is that we get journals like the European Journal of Taxonomy, which is PDF. We get service contracts and we convert them. So essentially we make a contract, we design what granularity or quality they want, and then we just process them. So you can read more about that in November. There should be a publication coming out in EJT explain this more in detail. And the other part is, I think, which is an important one, is there are some foundations out there like Arcadia who actually is interested to make this move happen. And so they pay for data conversion, like they paid us, and we liberated like 400,000 treatments over the last couple of years. And I think this may be another way to look at that also for you in South Africa, to maybe find a way to get one or the other foundation to look into this, to help to convert journals which are from South Africa, to make them accessible so they show up in GBIF. That means you have really the data, you can add them in your fauna and flora lists. Maybe, I don't know, there that may be one thing which could be, because the point is really we are living in a digital world now, and there's funding essentially to digitize this world. And I think we have something very important to say, because we have all this data about biodiversity. And it's an expensive thing to make the data accessible, but it's a very important data. And once done properly, it's open forever. And it speeds up research, it speeds up access. And so there is interest in this foundational world out there to support that. The trick is always find them and then get these proposals done. But I think at the moment, this may be one way to go.
Oh, great. All right, Peter. You're muted, Peter.
Yeah, hi. Hi, Donat. That's really excellent. And I really appreciate your making all this information more easily accessible, because especially for someone like me who's in my own private institution, I don't have such easy access to libraries and that. So it's wonderful to be able to find stuff far more easily than we used to be able to. One question I have on the really old publications. One of the tricky things there I've had a lot of experience with is trying to digitize something from 150 years ago and then translate it, and getting a clean enough copy to be able to actually translate, because I don't know enough French or German or Italian to do it myself. So I have to rely on online translators to help me. But to get a clean digital copy is really tricky. Is there any possibility that we're going to have the older stuff sort of digitized, OCR'd in a decent way that you can use, and then be able to translate things and make those translations available? Because at the moment I find, I'm sort of translating a description here, a description there, but it's very piecemeal. But if we could get even those bits made accessible to other people so they don't have to reinvent the wheel, or get entire publications translated, that would really also speed things up for a lot of people.
Yeah, you touched the point, the most costly point in this entire endeavor. It's to go to a library and get a copy, a digital copy, which is good enough, and then do all this translation. So generally, the older publications get, the less you use OCR, you need double keying. So you need some experts, like specialists, who can actually deal with these old fonts. So that's one thing we also look at with the Biodiversity Heritage Library. How can we actually make sure we can get this text out of these publications? And that's a widespread phenomenon, but there's no short answer to that. And the translation is an important one, because right now what we do, it's sort of agnostic of language. So we can extract modern publications in Chinese or in Arabic or so, because the structure is always the same, so you can do it. But translation is a different thing. And that it's an interesting point, because in the Cold War time, everything, like all the Russian journals, got translated into an English form. So there were like offices in Canada and then the US would translate all the Russian entomological journals. So we had like this translation service. So we can't, of course, not hope to get back in the Cold War, but I mean, it's a big thing.
Yeah, it is really tricky. As you say, the old fonts are the issue. It's quicker to literally sit and type an old publication in word by word than to try and use OCR.
And you see how complicated it is, because the second volume of Systema Plantarum from 1753 doesn't exist in a transcribed version yet. It's only the first volume, when nobody made the effort to translate this very seminal, important work into a way we can actually use it.
Okay, all right. Any more questions? I'm just going to ask one more. Oh, Nicola. All right, go for it.
Yeah, just another one. I think, Donat, you touched on this issue of duplicating work and overlaps with other databases. I mean, that must be also potentially quite a difficult thing to do, to work out. I just wondered if you could talk to us a little bit about that.
About what exactly?
About making sure that you're not duplicating work that other databases are doing, or that, you know, so that people can search in one place, and there aren't other people who are doing similar things.
Yeah, that's a complicated thing. I mean, the optimistic view is that if we expose everything, people will find it at some point. And we're not anymore in an age where well, still we compete a lot, but essentially we slowly start to understand that databasing is something you don't want to compete. You really want to have the data accessible so you can do really interesting stuff. That's not databasing and be owner of a database. But it's important to have it. So we are not the right person to ask about this, because that's a complaint about us that we are too quiet and people don't know. That's why I'm happy to be able to talk to you about what we do. And we will make, I promise we'll make an effort now to be more outspoken and talk more about what we do. But I see already like in GBIF that there were like 180 people sitting in the governing board of GBIF. And I think they're all aware now of what's happening in terms of data extraction from publications. So I think this word is spreading slowly. And data is also in GBIF. And the other thing which I think is really important to understand is that what duplication means. So processing the same article twice, that's duplication of work. That doesn't make sense. But creating duplicates material citations because they're cited in different articles are not duplicates. Or creating occurrences of the same specimen, multiple occurrences of the same specimen in GBIF are not duplicates. And that's important to understand, because if you have a specimen in GBIF, which is digitized by Zombie or somebody and sends it to GBIF, so there is a record of the specimen. If now somebody has a publication and cites this specimen, so we create a material citation, send it to GBIF, and suddenly there's a second record of this specimen in GBIF. Then GBIF starts to discover that, so they run what they call clustering algorithms, that they bring together all the same specimens in groups. And what it essentially does is that it says, okay, Zombie has been the first to upload a specimen as an occurrence to GBIF. So that's essentially the beginning of the history specimen in GBIF. And then somebody uploaded a sequence in NCBI. So this is about the same specimen, but it's a sequence. And then we come in and upload a material citation. So there's a next step, and it's okay, we added more, we augment the knowledge about this specimen by adding this material citation. So we're adding a scientist's view of an interpretation of this specimen. And it's important because this way we can build up knowledge about a specimen. We get to know everything we know about a specimen in your collection, who cites it, what sequence is known about it, and so on. So these duplications, in a sense, it's very, very important that we do it, that we try to liberate as much and make all this accessible.
Oh, okay, great. Yeah, so that's one of the things that I'm interested in, is this material citation stuff. So I mean, imagine if a herbarium could pull some metrics about how often the specimens are getting used and which specimens are getting used in publications. I think that would be very useful for museum and herbarium collections to have.
Go, if you have some time, or ask tomorrow, go into the treatment stats. That's our statistics. And it's a bit overwhelming, I guess, at the beginning, but it allows you to look into our raw data. So you can put in an acronym of your collection and see whether you find something inside. And if you find there are errors, because it's a lot of this automated, not QC. But the interesting thing is each one is linked to a treatment. So if you have a material citation, you can go and read the treatment of that. So you see essentially this is about South Africa, and this specimen or this species I'm supposed to be. So you have it.
Yeah, great. All right, I think we can probably move on then with the next presentation. Thanks, Donnet. Okay, so our next presenter for today is Thembile Khoza. Thembi, are you here? All right, and we're going to have a recorded presentation provided for this. So Thembi is the animal checklist coordinator with SASSB. I hope I've got that title correct, but she has the task of trying to make sure that SASSB is able to publish up-to-date species checklists or taxon checklists for all of the animal taxa in South Africa. I'm sure you can appreciate that that's quite a daunting task. Thembi is also interested in spider taxonomy, and she's done some work on, I can't remember exactly which group of spiders. But yeah, I think I'll hand over to you there, Thembi, to take it away.
Good day. Thank you for this opportunity. Today I'm going to talk about the compilation and dissemination of animal checklists for South African species. Just to give a background, the South African National Biodiversity Institute, SASSB, was mandated through the National Environmental Management Biodiversity Act, which was approved in 2004, to coordinate and promote the taxonomy of South Africa's biodiversity. In partial fulfillment of the mandate, SASSB embarked on an initiative to develop and update checklist of animal species of the country. South Africa has a rich biodiversity fauna with more than 65,000 animal species that are estimated to exist. In collaboration with partner institutions, taxon experts, a checklist of South African animal species is compiled and updated based on new publications. The animal checklist policy is also available, and the aim of the policy is to provide a standard guideline for compilation, updating, verification, and management of the South African animal checklist, as well as its update to ensure consistency, transparency, and credibility. The policy is reviewed annually by the zoological research scientist, led by the animal checklist coordinator, or upon request from the users, and shared with the species working group committee. We are working on the checklist because they form part of the value chain and they provide baseline information on valid and preferred species names. They also contribute in influencing policymakers and government's high objectives, such as job creation. In terms of data compilation, there are a number of research institutions, universities, museums, grant funding projects, researchers, students, and retired taxon experts who are still willing and committed in assisting and contributing towards completing and verifying the checklist. For the taxonomic backbone, we are following the Catalogue of Life classification, which gives the preferred scientific classification for each species. And these core fields are mandatory and must be populated for each taxon to be accepted. The metadata file is also included in the original checklist to indicate and keep track of the people who contributed to the checklist, especially the taxon experts who maintain the full ownership of the data, and they can be acknowledged accordingly. And for data verification, the checklist committee was elected, and it includes the members listed below. The main responsibility of the committee is to verify and approve the checklist before it's published. And for data dissemination, the Biodiversity Information Management team is currently working on the information system, NBIS, which is going to house all the data sets. Currently, the data inputs for checklist and the e-Fauna from FBIP and other projects is submitted to the Zoological systematics for data quality checks and inputs. Then the data will be submitted to the committee for approval. Once the data is approved by the committee, the information will be uploaded to Specify or ZooData, and dedicated members will continue with the data correction. The taxonomic backbone is currently running on Specify 7, and it includes a number of collections, especially the ones that has taxon experts working on them. For example, the arachnids, butterflies, mammals, birds, fish, frogs, and reptiles. Specify is also user-friendly, and it can also cater for a number of functions depending on what you want to do. And once your data is uploaded, we are able to add new taxa and modify the data accordingly. In conclusion, the Zoological systematics team is responsible for ensuring that the data in Specify or ZooData is complete, accurate, and ready for dissemination. And the Biodiversity Information Management team is responsible for ensuring that the system is functioning properly. And the Information Technology team is responsible for ensuring that the infrastructure is in place to enable BIM to effectively disseminate the outputs. We would like to thank the listed organizations, institutions, which includes researchers and students, for their contribution. Thank you.
All right, great. Thank you very much, Thembi. Just looking for... Can't seem to be able to, someone's got their mic off and I can't seem to be able to mute them. All right, so do we have any questions for Thembi about the animal checklist? All right, Chifiwa.
Okay, thank you, Ian. And also thank you, Thembi, for the information. That's quite useful. I just want to find out, with regard to the zoological systematics, who exactly are these people, or what is it? Can you please expand on that?
Okay, on the zoo systematics team, we have Pulusho Make and Ritabile Mutloung, who's responsible for the e-fauna, the species pages. So we only 3 members in the team. So we are mainly depending on the taxon experts and people from the university, the researchers, the students, and people who get funding from FBIP, so that whatever information they publish, the new species names, so we are depending on the universities mainly.
Okay, thank you. I get it.
Great, thanks. Nicola?
Hi, thanks very much, Thembile, for running over that. That's great. And I'm obviously more familiar with the plant checklist. And so, and I'm sure Ronell will talk about this a bit later. And so I'm familiar with the process that's in place when there's controversies in accepted names or taxonomies in the plant checklist. And I wondered how you deal with that. Do you also have a committee that meets to discuss specific issues, or how do you solve those problems?
Yes, we do have an animal checklist committee. The main challenges that we are facing at the moment is that we are only focusing on doing South African species. And then we have the red listing team, which they are doing red listing for Southern Africa and global species. So those are the challenges that we are facing now.
So you're not, if a genus has relatives outside of the Southern African region, those are not incorporated into your checklist, right? Is that what you're saying?
No, because for South Africa, we don't have a backbone for all South African animals. So the question is, do we do the global backbone, whereas we don't have our own South African animal species backbone? So the main aim here is to make sure that we have the backbone for South African species first.
Right. Okay. And what sort of stage is that now? I mean, do you have a complete checklist at the moment?
Yes, we do. We have covered about 95% of the species. So the groups that are remaining are the ones that they don't have taxon specialists that are working on them. So we are relying on the taxonomy that is available, the literature that is available. And the challenge is that some of the old publications, they are in images, and some they are in German, and we can't get to interpret what is written there. Is the language barrier that is the challenge. So hopefully tomorrow on the workshop, we'll be able to get how can we solve some of the problems.
Okay, so I'd like to ask a question then, but I don't think I'm going to be popular with Thembi after asking this question, because it might mean more work for zoo systematics. So Donat, would it be possible to get a list of animal species names from Plazi that have been published, that have been described from South Africa, or that have South Africa in their distribution information, or have material citations from South Africa that we could give to Thembi?
Yeah, we can look into this. Yeah. So if you process material citations, then we would know. And so this needs data cleaning, but I think it's probably an interesting starting point.
Okay, great.
So if maybe she will just ask tomorrow or send us emails and we can help to get this done.
Yeah.
Okay, great. So that's more work for you, Thembi.
And the other point, maybe you should also talk to, if you don't already, and I guess you do, and so excuse my question, talk to GBIF about a checklist for South Africa, because they have a lot of data which is not coming from South Africa, but about South Africa. So, and they are in the way now to build like hosted portals. And that's a new offer from GBIF. And that's essentially also allows to build like a website for South Africa.
Yeah.
And that it's based on the fact that you can extract all data, not only from, but also about South Africa.
Yeah.
And then I will go and download that and check all the names there. And what's also happening in the next year probably is that the catalog of life and the taxonomic backbone in GBIF, they will merge in an extended catalog of life, which includes like all the 30,000 catalogs we provide through publications as well. But that will give you even more to look through, and then you should be able to check like parts out and say, give me all South African material, for example.
Yeah. So another thing is that in your presentation, you didn't mention how do you deal about the species that has been synonymized, because even if you have all the data under one specimen, but if the species has been synonymized, so how do we deal with that?
I mentioned in the, and probably nobody realizes that, is that there's treatment citations. So even Linneo started doing that. So he said, okay, this specimen or this species have been cited by this author in a previous publication. And so what we do, we make use of that. So if there's a synonym, it's normally cited in a publication. So you have a section in the text. Normally you have like the nomenclature section, the first part, which is like normally a title or something like that. And then you have a section where you include all the synonyms, or you include like where this species have been described first time. So these are what we call treatment citations, and they're picked out. And this allows us then to take it out and make a relationship and say, okay, in this publication, the author says he cites Linnaeus taxon. So we have like a tool which is called Synospecies. I didn't show it today, but we'll show tomorrow, which essentially allows you to go through that and ask the question, what is the currently accepted name? And so if we the tagging rights, then the system will tell you this name is now this, or this has this and this other synonym of that.
Okay, thank you.
I mean, that's why I think it would be interesting to actually go through literature, because you have exactly this tagging, these citations and references are all in the literature. And so you normally go in and get publications and find out, okay, this has been synonymized. This you have a mental annotation in the text and say, okay, here is a synonym. Then you kind of discard this and then put this in your database and say, okay, this is a synonym of that. And then you say maybe in this publication. But with that, you break the link. So you don't know anymore where it is, because you don't have probably a link to a treatment. And the treatment is not annotated. So everybody else has to go back again and start from scratch. So if you actually do what we try to do to teach you tomorrow, make a little bit an effort to make not just mental annotations in the text, but also go in and make them physical. So you take the text, say, okay, here to here is a treatment citation. Then you do service to everybody else afterwards.
Yeah. Okay.
Great. Any more questions about animal checklists? Yes.
Yeah, can I just pop in? I think what we're getting at here is the difference between a sort of an old-fashioned static checklist and a dynamic one that's keeping up to date with the latest taxonomy. And ideally what you want to be doing is having that checklist, if possible, automatically updating as new taxonomic treatments come out. So if things are synonymized, it can actually automatically be updated, and those annotations incorporated into, a species, if two species get lumped together or one gets split apart. So that we don't have the situation where every 5 years we've got to try and reissue the South African checklist. be far more efficient if we can have it being continuously updated.
So the feeling here is that, not my feeling, but what we observe is that, yes, updating, if you have a new species, it should be automatically updated, especially if we can control whether a name is available or not. But these are things we can do by machine. We can find out whether the animal name has a description, whether it has a material citation, so it cites a specimen, which is called holotype, and somewhere written that it's in what collection. So those are what you need in an available name. So for synonyms, there are all these checklists out there, and they're very clear. This has to go through a human to be checked. So what's probably we're going to see probably is that increasingly we make everything automated, and in GBIF there will be 2 catalogs. So there will be the extended catalog, which includes all the automated stuff. Then there will be the Catalog of Life Plus, which is the new version of Catalog of Life. And if something has to go from the extended catalog into the normal catalog, there is a way that the experts can come in and say, okay, I agree on this synonymy or not, and this is actually the same name or this is not the same name. So it requires that they want this step. I mean, if you look at that, it's interesting because we have a problem about human resources to actually do all this cataloging. And so if you have this, if everybody, you have to go like thousands and thousands of publications a year, maybe 20,000 or so, then who's going to do that? So there will be very, very quickly, there will be a huge catalog of extended catalog, which is completely, more or less completely automated. And then you have this hand curated part, which maybe it's more reliable, but it's also much more incomplete.
Yeah, interesting. All right, have we got any more questions? So Nicola, yeah, you had your hand up.
Yeah, I think this kind of follows on from this, because it's, I mean, in the plants I work on, I will, you know, there's databases like IPNI and Q's got Powwow. And if you go onto those databases, you'll often see different treatments. And actually the most frequent one is that there might be alternative generic placements for things, and they just get shuttled between them. And it can be quite hard to know what the latest one is. And I think, yeah, I think exactly what Donat was saying, that if you link to a publication, it becomes clearer how to treat these synonyms. But if it's just a checklist, even a digital checklist, then that's where you can get problems and issues. And this also is kind of, I think what I also meant by duplication earlier, that you have, okay, so for daisies, we've got the global composite checklist, there's IPNI, there's Powwow, there's GBIF, Catalogue of Life, well, those two I think work together, and so on. And when they're in disagreement, yeah, that's kind of the duplication. I think that can be an issue.
Yeah.
I mean, making things digitally accessible is the best thing we can do, because it exposes all these duplications. And from a user point of view, normally you have to build up trust. That means you see, I believe more in this database or another one because this is more complete, or it does what you expect. And it's sort of an arbiter out there. The crowd essentially kind of in the longer term decides what is the one which will survive. But I think it's important we talk to each other and really make use of it. And that again is one thing which is often discussed now. This is, each catalog has a slightly different focus. Some need just names, other ones more focused on curation of collections. And so there are different they do these things different, not because they look at things differently, but because they have different purposes. So that's essentially important to understand all these different databases. Why is this database? Is it just to get a list of names, or is it to curate names or for what?
All right. Okay, so it seems like the conversation is going in the botanical direction now. So that's a good place to introduce our next speaker, which is Ronell Klopper. So Ronell is the coordinator for the plant species checklist with SASSB. So this is a national standardized species checklist for plants that we have for the country. And yes, she's also very much involved with plant taxonomy in SASSB as well. So I'll hand over there to you, Ronell.
Good afternoon, and thank you for this opportunity to tell you about two closely linked ongoing projects. First, a bit of background. The flora of Southern Africa region shown here in the map is a hotspot of botanical diversity. It harbors the richest temperate flora in the world and is home to 6% of the world's plant diversity, including an entire floristic region. 50% of the vascular flora of Africa, as well as 50% of continental endemics, occur in Southern Africa, which is less than 10% of the total land area of the continent. 50% of known succulent plants of the world occur in Southern Africa. The South African National Plant Checklist and the e-Flora of South Africa projects are collating information on our diverse flora and making it available in encompassing products to the benefit of end users. In this presentation, we will give a brief overview of these two projects, their history, some aspects around their compilation, and the outputs from these initiatives. Although the checklist and the e-Flora currently focus on South Africa, the history of checklists and floras in our larger region is intertwined, and I will try to make it clear when I refer to South Africa as the country and when I refer to Southern Africa or the flora of Southern Africa region, also known as the FSA region. The checklist and e-Flora covers indigenous and naturalized taxa occurring in South Africa. Plant groups that are currently covered in the checklist and e-Flora are the mosses, liverworts, and hornworts, the lycophytes and ferns, the gymnosperms, and the angiosperms. And we are also in the process of adding the marine macroalgae to extend the coverage of the checklist and the e-Flora. Currently, the checklist thus covers 381 families and over 2,600 genera, which comprises over 21,000 species, of which around 20,000 are indigenous, with a species level endemism of 60%. The e-Flora of South Africa has already accumulated descriptive information for 98% of indigenous species and 60% of naturalized species. Plant checklists and floras have their origin in lists of plants accompanied by short Latin diagnoses that were published in the 18th century. Although Southern African plants appeared sporadically in herbals and catalogues from as early as 1605, one of the first catalogues to include a large number of Southern African plants was that by Burman. Probably the first publication to focus entirely on South Africa was the first Flora Capensis, a 19-page book by Linnaeus on the 502 species that early collectors sent to him from the Cape of. In 1772, Karel Thunberg, a student of Linnaeus, visited the Cape of Good Hope and published numerous academic dissertations and other publications describing the plants that he had collected, of which over 1,000 were new to science. And Thunberg is known as the father of South African botany. Several taxonomic treatments of the Southern African flora followed, including the well-known Flora capensis started by Harvey and Sonder, as well as several volumes of the Flora of Southern Africa series that was published between 1963 and 2005. In the early 1990s, focus was shifted from completing this FSA series to producing regional floras or conspectuses to completely cover the flora of Southern Africa, and the following volumes have already been produced. The Northern Provinces, the Fynbos region, the Succulent Karoo, the Free State, the Free Volume Eastern Cape Flora, and only the Nama, Karoo and KwaZulu-Natal remains to be done, and these are nearing completion. So numerous other works were and are still being produced to describe the collections of Southern African plants, and this information is sometimes consolidated into monographs, field guides, taxonomic revisions, and other articles. Computerization of information from these numerous publications started when the Plazi database was developed in the 1970s. It housed data from specimen records from the National Herbarium in Pretoria and also had a taxon component for the checklist. Over the next few slides, I will illustrate how the checklist developed over the years and how the information associated with the checklist was expanded towards providing a complete e-Flora of South African plants. So the first output from Plazi was published in 1984, and it contained a simple list of accepted names of Southern African plants. It was followed shortly by a second edition in 2 volumes that also included synonyms and references to recent literature. And updates to the checklist were regularly published in the journal Bothay. The third edition of the checklist contained regional distribution as an additional feature. And information in the 4th edition was expanded to also include data on life cycle, life form, height of plant, and altitude. So these were all regional checklists for Southern Africa. And the first country-level checklist for South Africa was published in 2006. The 2003 edition of the Southern African Plant Checklist was made available online on the Plants of Southern Africa, or the PAWSA website, in 2006. This website initially contained the same information that appeared in the publication, but 2 website upgrades saw the addition of plant images and distribution maps, as well as information and text from the FSA volumes. And updates to data were made on a regular basis up to February 2013. During this time, data from Plazi was migrated to a Brahms platform, which is a different type of software that runs the database. And during this process, the database was renamed to the Botanical Database of Southern Africa, or BODATSA for short. Following migration, the PAWSA website was also redeveloped and relaunched in 2016. For the first time, the website also included specimen-level information from collections housed at the 3 SASSB herbaria, thus aligning with global efforts to mobilize plant biodiversity data. Data on the website is updated at the beginning of each quarter. Also available on PAWSA is an official yearly release of the South African Plant Checklist as a downloadable Excel file, which is released towards the end of January each year. It is also disseminated on SASSB's institutional repository, together with other checklist-related documents like the checklist policy, the classification system followed in the checklist, and annual reports on changes and additions made to the checklist. As a next step in the process, the e-Flora of South Africa project has expanded the online data provided with the South African Plant Checklist. As a participant in the global strategy for plant conservation, South Africa committed to produce an electronic flora for the plants of South Africa as part of efforts to reach Target 1 of the 2020 GSPC, namely to produce an online flora of all the plants of the world by 2020, which will continue into the future. The e-Flora of South Africa is able to contribute 6% of the world's floristic data to the World Flora Online project, which is hosting the e-Flora. The e-Flora of South Africa accumulates published data through an aggregator portal method. Everything starts with the checklist, which is divided into smaller taxon units, usually genera, and for each group, relevant literature with the descriptive information that we want to include in the e-Flora is selected. Permission is obtained from copyright holders to use the information. Literature is digitized where necessary, and text is marked up for further processing. For this step, the taxon markup. This is a plug-in for MS Word, and one starts with the digitized literature that was converted into a Word file. Clicking on the Taxon Markup tab will open the ribbon that contains certain features, the most important of which is a box to display the Taxon Markup panel, containing fields for the taxonomic and descriptive data that is used in the e-Flora. Markup is done by selecting relevant text in the literature and transferring it to the relevant field. Navigation buttons allow you to create a panel for each taxon in the publication and to navigate between these panels. Once all information is marked up, the information is extracted to an Excel spreadsheet. Data from this spreadsheet is then imported into Budatsa, and from there it is published online. If you want to know more about the workflow and processes of the e-Flora of South Africa, these are explained in more detail in this publication. The e-Flora of South Africa project will provide species pages that contain descriptive information. This will be made available through SASSB's Biodiversity Advisor website, which is currently being redeveloped. And herewith, I give you a sneak preview of what a species page will look like. But unfortunately, there's not enough time here to explain these features in detail, but please space. Just to summarize why we need the checklist and the e-Flora. A national species checklist is an essential resource forming the taxonomic backbone for all foundational and applied research and biodiversity-related activities. It is crucial for herbarium curation and where CITES and other legal lists depend on correct nomenclature, classification, and identification of plants. Information from the checklist and e-Flora affect decision-making and policy, for instance in the drawing up of conservation and development plans, and in red list and ecosystem assessments. The checklist and e-Flora also feeds into global initiatives such as the African Plant Database and the World Flora Online projects. It is thus essential that information in the checklist and e-Flora is as up-to-date and accurate as possible, and that this information is widely disseminated. But how is all of this kept up to date? Firstly, there is a clear policy and strategy that guides how updates to the plant checklist are done and which groups should be prioritized for updating. 2 staff members with a sound knowledge and understanding of plant nomenclature and literature take full responsibility for maintaining the plant checklist. Only published changes are incorporated into the checklist, and where competing published classifications exist, these are referred to the South African Plant Checklist Committee for consideration. This committee is comprised of representatives from several South African institutions who are qualified taxonomists and active in the field of plant taxonomic research and/or herbarium curation. Changes and additions to the checklist are filtered down to the e-Flora project, where descriptive content for new species are continuously added and other information expanded and amended to reflect new taxonomies and classifications. All of this is currently a very manual process, and we hope that the Plazi initiative and how they make digital taxonomic literature available will help us to streamline our process in future and add a more automated component towards maintaining and expanding the plant checklist and the e-Flora of South Africa. Thank you for listening.
All right, great. Thanks very much, Ronell. That was very interesting. Once again, we'll go to questions. Hands up first. All right, doesn't look like we've got anybody rushing for questions here. Nicola?
I don't have a question. I'm very familiar with this. I just wanted to thank Ronell for giving us such a clear history and explanation of the checklist and the e-Flora and its sort of relevance. Yeah, thanks very much.
Thanks. Benny?
Yeah, I have a more of a philosophical question, actually. It's already quite difficult in a South African context to come to an agreement on which taxonomy we are going to follow. I'm very much aware that we have a system for deciding which taxonomy for South Africa is the best. We get to a compromise, and that is the compromise that gets into the checklist. Now, we are more and more aiming for a world checklist of plants, which essentially means that we are going to have only one opinion on taxonomy, on the taxonomy of certain plant groups, that is going to be propagated. And it could well be that the opinion of South Africa is not standing up to opinions from, I would call it, even more powerful institutions. So I'm a bit worried in the long run how this is going to pan out. Is there really a politics of big institutions that are able to push their taxonomic opinion forward? And is it not going to be that certain countries which have less powerful institutions, and we are still, I would say, reasonably off, but take smaller countries, what are they going to be able to bring to the table in terms of saying, this is our taxonomy, these are groups of plants that occur here. How will we push our taxonomy, the one that we prefer, forward? And it's all right to say, listen, we look at the latest publication, but it's also very easy to find contrasting publications based on the same data, because ultimately the decision as to what is a genus and what is a species is a human decision. So the interpretation of phylogenies can be very, very different. And I'm afraid a little bit that in the long run, we are going to an undemocratization of taxonomy. I'd be curious to hear Ronell's opinion on this.
Thank you, Benny. Yes, that's indeed a very, very big problem that we are facing. But we are trying to stay current and involved in these global initiatives. Like I say, we feed very much into the African Plant Database and the World Flora Online, where we try to make our opinion heard, especially concerning our endemic and near endemic plants, because that's the plants that we know and we know what's going on there. So we try to make sure that our voice is heard, especially for those plants. We already follow a slightly amended family classification for the angiosperms Where most other institutions follow APG4, we have amended that slightly, where we still recognize some of the bracketed families from APG2, which are in many cases have their centers of diversity in Southern Africa. So for our plants, that family classification that we have decided upon amongst various taxonomists in South Africa, we've discussed it widely, and everybody concurred that it makes sense to rather use that than the APG4 because it makes more sense from our perspective. But we do still need to make sure that we can translate it into an APG4 classification. For instance, if we provide data for the World Flora Online, because that's what they follow in terms of families. But for genera and species, especially where it's our endemic or near endemic flora, I think we need to make our voice heard, and we are trying to do that, especially through the World Flora Online. We are in the process of adding a large number of our endemic plant names and their synonyms. That's not, some of them are not even in IPNI, some of the synonyms. And to add them to the World Flora Online backbone, with South Africa being sort of the taxonomic expert network, if there's not already one for that family in the World Flora Online project. So we are definitely aiming to make sure that our voice is heard, whether it's going to be shouting loud enough from down on the southern tip of Africa so that everyone can hear us, we're not sure.
If I may add a short word on that, one thing I like very much in the African plants database is that it actually shows who agrees and who disagrees with a particular system. And there are very few databases that present the data in such a way whereby for a particular plant name, it is said accepted in South Africa, but for instance, rejected in tropical Africa. Do you think there is room for that in a world checklist?
I would hope that there is, and that's one of the things that we were very adamant about when we combined our Southern African data set with the tropical African data set of Genève and to create the African plant backbone, that we didn't want to sort of force an opinion and rather just say, here are a difference of opinion. And that also sort of highlights taxa where further work is maybe needed. So I think definitely that should be considered in a global checklist. There's more and more initiatives and talking into that direction. In 2019, there was a big meeting in Geneva where several role players from GBIF and Catalogue of Life and World Flora Online and several others were present to sort of start talking about a consensus checklist. And I think this issue, if I remember correctly, was also raised there, that there are certain places where people will not agree and that one should sort of, you know, a classification is a hypothesis, not either one of them is correct or wrong if they're based on the same data set. So you can choose whether you want to follow the one or the other. This is something that not many people understand. But there's definitely scope for that option, I think. Even in a consensus checklist, there should be sort of like a flag, alternatively treated as this or something like that. I think that's definitely something that one should keep considering. Thanks, Penny, for raising that.
No, so maybe if I could just follow on from that is something that I think is going to become increasingly important is the use of actual taxon concepts. So when people use taxon names, they specify the actual concept that they're referring to. So this is something that we're trying to push through the NSCF. So you have, this is the name, but I'm using this particular author's concept of that taxon based on that name. And I think that's from the sort of bigger biodiversity informatics perspective, I think that's going to be quite important.
Yeah, that's something that we try to do in Bodatsa. There's a function where you can link a name to literature, the taxon literature link, where we will then say in this literature on that page, it's treated as accepted or as a synonym. And then obviously that sort of, it's not quite, it's the same that you are talking about, but it's sort of moving into that direction that at least we have those literature links to the taxon name And if we treat it as accepted and we know that it's accepted in that publication and that's the latest treatment that we follow, because we also, for each genus, say this is the treatment that we follow, then at least that taxon concept based on that publication can be fixed or referred to, which I think it's important that taxon concept issue one should definitely keep that in mind. And it's also a bit of a new way of thinking about it.
So if I could just jump in, I think for a checklist point of view, the more important thing is your species concepts. And you're worrying too much about the higher classifications really it isn't worth wasting too much worry about, as long as it's specified what you're following. The real issue is whether we're keeping the species concepts as up to date as possible. So it would be nice if we had a unified global phylogeny of life that was perfectly based on evolution and DNA, but that's not going to happen for a long time. So I think we just accept the differences and try and make sure we have a correct list of the species.
I mean, one thing I think
absolutely
seems to be
agree,
interesting is if you can actually link like the concept or its treatments to specimens. So, you know, this person looked at these specimens which formed his opinion. I think that's one chance we have is in this digital world now that we could link this. So when you publish, you should say, okay, this is my basis. So you can go back and look at that. You could just essentially draw a map, put these things on a map, and you see, do we talk about the same or not? And then, of course, you could go back and look at images and so on. And in botany, I think that's quite possible.
Yeah, so maybe we should organize another SASSB session talking about taxon concepts. That might be a good topic. All right, Nicola?
Yeah, that sounds like a good idea, Ian. It'll be a very long session.
Taxon concepts, not species concepts.
Yeah, well,
I refuse to get involved with species concepts.
But that's been sorted out. Just to support, I think Donat's suggestion is really good, because actually, if you can link to specimens, then there's actually often a way through controversies. Potentially, I mean, that's a way that these new tools can maybe help us to move forward. So I think that's a really great suggestion. But then I also just wanted to make a point that I don't think this work is ever going to be done and dusted. I mean, now, you know, there's always new tools being developed and new ways of looking at the diversity out there and the way it's organized and the gaps in characters and new characters to look at. And I think it's actually the rate of change is actually going to ramp up rather than slow down, even though we get more. That's my perception of it. Even though we get more and more on top of it, our level of investigation of genes and genomes and probably even phenotypes, I think, is just going to ramp up. So I think all these things are really necessary and will become increasingly relevant going forward.
Oh, great. All right, have we got any more speaker questions? So I'm going to assume not then, and we're sort of approaching the end of the 2 hours with about 15 minutes left. So what I thought is, if there's anybody who just wants to, if we just round the session off with anybody's sort of own personal ideas or experiences with anything related to digital taxonomy, working with any kinds of databases or anything like that. If they just want to, if anybody would like to offer that in, offer any thoughts or opinions or experiences, especially working with all of these tools that exist online, not necessarily related to anything we've discussed today. I'm inviting contributions from other members of the group who've been less vocal today. That's what I'm asking for. Yes, Benny.
I will say something more than I've just gone through a course with my honors students on digital taxonomy or digital tools for taxonomy. And I'm getting questions often from colleagues or even from amateur botanists, citizen scientist botanists. And it's surprising how much misunderstanding there is about what these databases can do. I very often get the remark, yeah, but I found this name on IPNI. So people assume, because they don't know, that names on the International Plant Names Index must be accepted or correct names, while that is not the function of IPNI. IPNI is just a list of all names that have ever been published. So we also need to do some effort, I think, in trying to explain what the function, what the use of certain databases is, and what you should also not use certain databases for. And that's my experience. I've learned that in the hard way, that there's lots of misunderstandings. The tools are fantastic, but they are sometimes abused or misused, and we need to do some effort in explaining that to colleagues and to the general public.
Oh, that's very interesting. So I think something else with these sort of big online digital databases is that they can be very intimidating for people. So, you know, it's big fancy systems using fancy technology, and for a lot of people who aren't sort of that, you know, that comfortable with that kind of technology. I mean, you know, Donat was showing us JSON data sets and XML data sets and things like that, you know, which is quite sophisticated stuff. And so it can be quite scary, but I hope that it's something that that we can start sort of trying to address in the community here in South Africa, is developing sort of greater levels of competency for working with these systems. Has anyone else got any experiences they'd like to share?
I could pop in a little punt here for looking at AntCat. If you want to see the kind of database that tries to show the completely up-to-date taxonomy and taxonomic history of every name. So that's being run mostly by Brian Fisher, but it's based very much on Barry Bolton's Ant Catalog. And that gives you right from the current names right back to the protonym, and the actual type specimen is the aim, to have every name linked back to the original type specimen, and the full taxonomic history with every publication that's ever dealt with the taxonomy of a species linked. But it is a hell of a task. Keeping that up to date is pretty much a full-time job, and that's just for one family of insects. So the dream would be to have that happening for all taxonomy on Earth. Practically, I don't know that that's ever going to be feasible unless it can be automated through things like Plazi, pulling out the changes automatically from the new publications. That's why I was asking about, you know, whether you could get the synonyms and things like that just automatically updating. If those taxonomy publications can be formulated in such a way that the data can be pulled from them and automatically update something like AntCat. But certainly if you're involved in the checklists and trying to maintain up-to-date taxonomy, it's worth having a look at how AntCat is looking at doing that. I think, Donat, I think Brian is using Plazi a lot as well, as far as I'm aware. I don't know if you have any comments on how that fits in with AntCat.
Yeah, I think AntCat at the beginning was based on all the scan materials we had. So, and we also it was to some extent a parallel project, the Hymenoptera Online, which includes all, essentially, it's a complement to, or it's essentially an overlap between ant cat and all the Hymenoptera, which is much bigger than the ant cat. And it's interesting because the history behind it too, because it shows also a bit the fractions in this world. So ant cat has been one fraction, and the Hymenoptera Online was another one, and we were not really talking to each other. But we essentially started to make all the literature accessible. So we got grants from the Smithsonian at that time. So it was a really good base to do exactly what Bolton did, just to go through each publication and extract what you can. And so for a while, we worked on that to make, to essentially extract all the ant literature and then go through, but we did not get the funding to go through all the ant literature and make all these annotations. We originally started to do all the Madagascar literature. So this was our first. And if you go into Plazi, you can find this very good coverage for all Madagascar. So everything at that time, about 5 years ago, everything was in Plazi. But it's also been a good example that what it means that if you would work together, you could do much more. So if the ant community would work more together, then we would have everything. But because of these fractions between different groups, it kind of had some overlaps. But you also see that at the end, it is a huge amount of work to be done. If you get into this level of the ant cat, I mean, there's a huge amount of things to be done. And that's why I think really we should make really an effort to catch up, starting with new publications, extract everything we can automatically. So it can go in, and then people like Bolton or whoever, they don't have to search for literature, but they get like an interface where you actually see, okay, this came in today. Then you can go and you have an editing interface. You can say, okay, that's actually right, and this is not. And that's an interesting IT issue here is that you can provide values and say, this is trustworthy data, or this is kind of not so trustworthy, because the algorithms, they can find out. So if it's whether it includes what you want, or whether some things are missing or don't correspond, but it would be important in that too, that if like the plant list or so, if you start to work together, actually learn from each other, because one of the best thing is to improve systems is to learn from errors. So we all are completely worried about errors, but for us, errors are actually interesting, because if you start to analyze them, then you find out main error sources of errors. And then you can find out, is it because it's publication? Is it because of the algorithm? Or why do we have these errors? And one of the main error in publishing, and especially extracting material citations, is that publications, they are just texts. And the texts are just a long string from A to Z. Material citations, they are discrete units. And from an IT, from artificial intelligence point of view, it's extremely difficult to find out the begin and end of a material citation. And once you have the material citation figured out, then everything, it falls more or less into place. But if you miss a little bit, then things become complicated. And so that's why we started talking to publishers like EJT or Pensoft, and we published like lessons learned, essentially. These are like guidelines, how you should publish, so you can at least make things machine readable more. So it's clear we will not publish XML tomorrow, most of us not, but what we could do, we could, for example, between each material citation, add a symbol which is unique in the entire text, like a bullet or something. So when you actually process this automatically, you know, okay, here's begin and end of something. And that raises your hit rate by probably 90% or so.
Oh, interesting. Yeah, Nicola?
So Donna, in response to that, I mean, is there take up from publishers for something that simple? I mean, it seems so frustrating because, I mean, I started my material citations in an Excel spreadsheet, then I have to turn them into text for the paper, and then someone has to mine them out with great difficulty. And that just really seems, yeah, like a lot of work. But yeah, would publishers tell me that I have to put in a bullet point or some other symbol? Because that would be the way to make sure it happens.
Well, it's another job you need in your company, going out and talk to publishers and talk to authors to adopt that. But it's important, I agree. But I think it's a bit of a bold thing, but I think we should include, like in the botanical code, the recommendations that only a name is only valid if it's in GBIF. So essentially, you can define what is an available name. What does it mean? So it needs like a proper name of all this, but it needs a publication. It needs a diagnosis, whatever. It needs a holotype. It needs a collection where it is. But this is all things you could produce by machine. And if you actually would make this machine readable, and it shows up in GBIF, then you have solved essentially a lot of the issues we have now. It's a bit a radical thing, and I guess we should submit this recommendation into the botanical community. But I think essentially this should be done because that solves a lot of the problems, and it also frees time because IPNI will not have to go through its publication, extract things, and make a decision whether it's there or not.
I think that's actually less radical than some of the suggestions that have been passed recently. I don't know. Some of the other suggestions.
Ronell?
Yes, this notion that Donat brought up has been discussed widely at nomenclature sections in the past few congresses. And there's definitely a push towards something like that, where there should be a registry where a name should be posted or registered. I know the fungal community already does that. So it's definitely something that we will probably see happen in the future. It's just more a matter of which repository are we going to use at this stage. But I'm sure in the near future we'll see that as part of the code.
All right, folks, I think that's a good place to draw it to a close. I'd like to thank everyone for joining and participating and for sticking it through to the end. Once again, a very big thank you to all of our speakers today, to Donat and to Thembi and to Ronell. I think it has been a very informative session, and I'm looking forward to the workshop tomorrow for those that are joining for the workshop. We'll see you all there. And to the FABI team for hosting us, thank you very much. And to the SASSB team as well for your collaboration. That's been really great collaboration and support on this. All right, thank you very much, guys.
Thanks, Ian. Yeah, thanks for being host chair. Thanks,
right.
everyone.
Yeah. Thank you.
I know.
Thanks for giving a chance.
And thanks, Donat.
So we can end it. Yeah, I guess we don't have to, if we don't end the work,